Hello, and welcome back to Introduction to Genetics and Evolution. In the previous videos, we talked about the general principles of natural selection as applied to single loci or as applied to phenotypes. What I wanted to do in this video is to present to you some elegant case studies and examples of natural selection. Cases where, looking at animals or plants in the field, we tend to see these observations that seem to be best explained by the action of natural selection. So let's go ahead and get started. I have three general categories of things that we'll be talking about. First, we'll look at some case studies of mimicry. Mimicry, as you may know from the term itself, suggests imitating something else in appearance. We'll look at variation in space and time within a species or across species. And we'll look at the principle of sacrifice for family members. Why does it happen? Evolution by natural selection isn't always ruthless, as some may have you think. So, looking at mimicry to start with. Mimicry, again, it just means that organisms evolving to resemble one another. Right? Now, when can this be adaptive? The simplest time when this would be adaptive is if the model has some sort of warning coloration. Basically, if you want to look like something that is scary, that makes it so other things are less likely to threaten you or to eat you or things like that. So if what's referred to as the model, the thing that you're evolving to look like, has a warning coloration, and is actively avoided, then your appearing like the model species will make it less likely that you will be harassed or eaten. I think this is not a particularly good example here of a dog trying to look like a skunk, and it's clearly not evolutionary. But nonetheless, let's look at some real examples. There's two general types of mimicry. The first one we'll talk about is called Batesian mimicry. This is the case where the mimic is not dangerous, but the model is dangerous. In this case, again, it's useful for the mimic to look like the model because others will fear it or avoid it for whatever reason. So we have here the example of the non-venomous king snake, which, as you see, looks very, very similar to the venomous coral snake. We have an example here of a fly that looks very much like a wasp. And the wasps, of course, can sting you, and that's very painful, and therefore they're typically avoided. This fly, however, has no stinger, but it looks so much like a wasp that it tends to be avoided, and therefore its fitness is higher. In both of these cases, they are more likely to eventually survive and have more offspring because they're less likely to be harassed and or eaten. And this is a really elegant picture. This is a caterpillar that, as you see, looks a lot like a snake when it puts its rear up like that. So this is Batesian mimicry, the case where the mimic is not dangerous but the model is. Let me show you a different one. This is Mullerian mimicry. In this case, this is a lot of dangerous species, or multiple dangerous species, evolving to look like one another. In that sense, they derive an advantage from each other. So what we have here, these are all, the top row here is all from the species Heliconius melpomene. This is, a nice tropical butterfly. The lower ones are all Heliconius erato. But what you see here is this melpomene looks a lot like this erato. This melpomene looks like this erato, etc., etc., etc. Now, importantly, why am I showing you all these different pictures? In each of these cases, the vertical group here are from one local population. So in one particular population, the melpomene look like this and the erato look like this. In another population, the melpomene look like this and the erato look like this. It's very dramatic that within each of these local populations, they've evolved to look like each other, even though they are different from their own species in other populations. And again, in this case, they derive the advantage from each other. Because imagine if a bird eats a Heliconius erato and it has a bad digestive reaction to it. It's less likely to eat any more erato, but it's also less likely to eat any melpomene. They all derive this mutual advantage. In the case of Batesian mimicry, the advantage is only to the mimic. In this case, the advantage is for all the models to come together. It's a really elegant case of natural selection, don't you think? So, moving on. I want to talk a little bit about variation across space and time. Now, there are a couple of things that are often referred to as ecogeographic rules. So these ecogeographic rules are patterns of variation either within a species or across a species group that tend to correlate with geography. So for example, having a particular trait in cool environments but not having that trait in warm environments. And when you see this pattern over and over again, either within species or across species, within a particular species group, that's indicative that there's some reason behind this, that there's some adaptive value to having a particular trait in the cold but not in the warm. So one of these is referred to as Bergmann's rule. This was named after the German biologist Bergmann in the 19th century, who observed that animals in general tend to be bigger in cold environments or in high latitudes. Now, there's classic work on this in house sparrows, but it's been seen across and within various other species groups too. Now, why might this be? Well, again, as you're larger, right, you're increasing your volume quite a bit, but you're not increasing your surface area at the same rate. So therefore, you should be better able to retain heat. Now, this is just one possible explanation. We're not positive that this is the cause for it, nor are we positive in every individual case that this is a result of natural selection. But we see this pattern over and over again, and it's suggestive of natural selection, and it makes sense. So we'll come back to this sort of principle a little bit later on in the class as well. So Bergmann's rule, again, is that animals tend to be larger in cold environments or in high latitudes. A similar rule is Allen's rule, is that animals tend to have shorter appendages in cold environments. Now, why would you do that? Why would you have shorter appendages in cold environments? Again, think of it in the context of surface area, that if you have long, lanky arms, right, then you might be losing more heat in a cold environment. In contrast, if you have short, little stubby arms, then you're not going to be losing so much heat. So one classic example of this that's been cited repeatedly is in the context of the ears of the polar bear compared to the ears of other bears. Again, shorter appendages in colder environments. Now, interestingly, we see this with respect to bills or beaks in birds as well. So beaks and bills, as you can imagine, do lose some heat, and this is a thermal graph to show you that they actually do have heat within them that can be lost. And this graph here on the right shows various different species groups. So there's the lines in the graphs, various different species groups, and in each case you tend to see this correlation with latitude, where at higher latitudes the bill size tends to be smaller. At lower latitudes the bill size tends to be larger. Again, consistent with what you would expect from evolution by natural selection. The last one that I'll mention here, though there are others as well, is referred to as Gloger's rule. The animals tend to be more heavily pigmented in high humidity. Now, this is generally or typically true near the equator, and this has been observed particularly in various bird species such as song sparrows. In those cases, it's possibly related to bacterial activity, though again, we're not certain. But again, this is another ecogeographic rule where we see it over and over again. It's been suggested to be true potentially in humans as well, but again, the evidence is not perfect in this regard, so we don't know. Nonetheless, all these ecogeographic rules are cases where it looks like the action of natural selection has been present because we see these same patterns over and over again with respect to geography. Now, you wouldn't expect this by random chance. Why would it typically be that the cold environments have the larger animals just by chance? You don't think that's true. Now, I'm talking about this so far with respect to phenotypes, but you see the same thing sometimes with respect to alleles in cases where we can look at the alleles directly. Drosophila pseudoobscura has a set of alleles on its third chromosome, and in this video I'll focus on one that's referred to as arrowhead, or in this case abbreviated AR. This is a species that I actually conduct research on as well, but we'll be talking about the classic work by Dobzhansky from the 1930s and 40s all the way through his passing in the 1970s. One of the things that Theodosius Dobzhansky observed was a correlation with altitude, not latitude this time, but altitude, how high population is, and the allele frequencies of arrowhead. So one of the places he studied was in California, and he found that at low altitudes, like here in Jacksonville, arrowhead tended to be pretty rare. But as you went higher and higher in altitude, you tended to get more and more of this arrowhead allele. Interestingly, now he did some laboratory experiments where he varied the humidity, and in those cases, again, as he changed the humidity to match what you would observe in low altitude populations, arrowhead became rare. When he changed it to match high altitude populations, it became more common. And the same thing also happens with respect to latitude. So this is quite striking, and where all these different sources of evidence are supporting the same idea that there is some selective benefit to the arrowhead allele at environments similar to what you would see in high altitude or in high latitude. Now, this was true with respect to geography, but remember I said before this was space and time. You see the same thing looking at over time as well. Remember now, Drosophila species are, they breed very quickly. Drosophila pseudoobscura has its generation time in the lab of 3 to 4 weeks. So you have many generations over the course of the year. So there's lots of opportunity for natural selection to change the abundance of alleles over seasons. So he collected Drosophila pseudoobscura from this place called Mather, California, just outside of Yosemite National Park, in 1946 and 1947. And what he found was midsummer or early summer, he tended to find a lot of arrowhead. As you got later into the summer, as it was warmer, you tended to see a decrease in arrowhead. So you see this a little bit in 1946, but you see it much more strikingly in 1947. That arrowhead gets less abundant as it gets hotter each year. Remember, this is slightly high altitude too, so that's why June is actually still a little bit on the cool side in that particular area. So again, the abundance of the arrowhead allele decreases from spring to fall each year, and then it comes back the following spring. Very dramatic. So this was again a case of variation across space and time, consistent with the evolution by natural selection. The last principle I wanted to briefly touch on was this concept of sacrifice for family members, or what's often referred to as kin selection. So why does this happen? Why do you sacrifice yourself to help your family members? Or why do some animals do this? They'll often sacrifice to help others if they are relatives. Now, Darwin, as with most things, had some ideas on this, and he pointed out in his Origin of Species book, selection may be applied to the family as well as to the individual, and may thus gain the desired end. It's important to remember that you're related to your family. So when you help your family, you are actually helping the spread of your own alleles too. So let's look at this with respect to some pictures and stuff. So let's say this is you, okay? This is you there, and let's say that generally speaking, if you just were selfish and helped yourself, you would have 2 kids. And let's say then on average you pass half your information to each daughter, and therefore your alleles live on. You know, half goes to this daughter, half goes to that daughter. On average, you've reproduced yourself. There may be particular alleles that were lost, or some that you have that are now in 2 copies in the population, but on average it's passed on. Passing on nothing is clearly bad. You know, if you were just to sacrifice yourself for nothing, clearly that's not advantageous. So let's look at this third option. Let's say that now you have 4 sisters. Now you share half your genetic information with each sister, right? Assuming you're diploid, which I assume you all are who are watching this. So each of these has already half of your genetic material, okay? Now you didn't make your sisters, they were made by your parents, but let's say they're there. Now let's say each of your sisters has the potential to be selfish, and they're going to have 2 kids each. With each of these nieces, you share 1/4 of your genetic information. So this one has 1/4 of your genetic information. This one has 1/4 of your genetic information, etc. So let's assume that your help is critical to the reproduction of your sisters and to the survival of your nieces. Okay, and you have the choice now. You could either help these 4 sisters produce their kids, or you could have your own kids. Which one is actually better for you? Well, counterintuitively, if you help 4 sisters and die in the process, your alleles live on twice as well as if you had kids yourself, right? Because in this case, you've passed on 2 halves, 2 times 1/2, so you've passed on a total of 1 copy of you. Here, you've passed on 8 times 1/4, or essentially 2 copies of yourself. That basically your alleles live on better because you helped your sisters than if you were selfish and helped yourself. Now, there were some assumptions in this, but nonetheless, we do see this concept that sometimes it will be beneficial for you to sacrifice yourself, or for an organism to sacrifice itself to help relatives reproduce. Now, again, this comes back to Darwin's quote here about selection may be applied to the whole family. And the term for this is kin selection. And it's the evolutionary strategy that favors the reproductive success of an organism's relatives, even when at the cost of the organism's own reproduction. There's a famous quote by J.B.S. Haldane, an evolutionary biologist. Now, he was asked if he would give his life to save a drowning brother, and he said, "No, but I would save 2 brothers or 8 cousins." Now, of course, it's a little bit silly, but you see where this concept is, that if he saves 2 brothers and each of them has half his genetic information, it's basically the same as saving himself. You may not feel like that with respect to your brothers, but that's at least what natural selection would favor. Now, there are some possible examples of kin selection out there. Now, one of them is with respect to the Belding's ground squirrel alarm calls. Now, these squirrels will, when they see a threat, will stand up on a rock or something and make a little chip sound. And it's potentially dangerous to themselves that they're doing this, because they're making themselves very conspicuous to whatever the predator is or whatever the threat is that's imminent. Now, it's thought that this may have evolved and spread to help relatives. And the reason for that is that females do alarm calls more frequently when there are relatives around rather than when there are no relatives around. And this is for one particular type of alarm call. So again, this is a possible example of kin selection. So again, overall, what I wanted to do in this video is just give you some case studies of natural selection and areas where it's been applied to some interesting natural observations. First, with respect to mimicry, so the resemblance of something that is a threat. Looking at variation across space and time, where often it's, you see this repeated observations suggestive of the action of natural selection, and of course, sacrifice for family members or kin selection. Well, I hope you enjoyed this. Thanks so much.
Hello, and welcome back to Introduction to Genetics and Evolution. In the previous two videos, we talked about the effects of genetic drift over single generations and over multiple generations. Just to stress some of the points I raised there, in single generation, genetic drift is about equally likely to make alleles increase as decrease in frequency. Okay? Over multiple generations, genetic drift can be somewhat predictable in that we expect the probability of eventual fixation, eventually getting to 100% for any variable allele, is equal to the frequency of that allele in the population. What we're going to do now is look at the effects of genetic drift on the rate of neutral molecular evolution. And this is something that's fundamentally important and will tie in with our next set of videos on molecular evolution. Now again, are the long-term effects of mutations in genetic drift predictable? Well, we've already talked about mutations arising at some rate, that there's some predictable rate at which new mutations can occur. Right? Now, some parts of the genome will get mutations that have no effect on fitness, right? Or some mutations may arise in a part of the genome that can have an effect on fitness, but the mutation itself doesn't affect fitness. In these cases, these mutations are referred to as neutral if they have no effect on fitness after they arise. They can spread by genetic drift, or they may be lost by genetic drift. Now, the question is, can we predict the rate at which they both arise and spread to fixation? This is what ultimately will lead to differences between species, right? That a new mutation arises, it spreads in one lineage, gets to 100%. That makes that lineage different from other lineages because it has this unique variant there. Well, can we determine this rate at which they arise and spread? Well, the tricky part is the ancient population sizes are unknown. So it makes it seem like this would be very challenging. Well, let's break this up into pieces. So mutations are arising, and let's say they arise at a rate which we'll refer to as mu. Right? This is the Greek letter mu. So this mu can be measured perhaps as mutations per year or as mutations per generation. We'll focus primarily on the per year side of this video. So let's imagine that our mutation rate is 1 times 10 to the minus 9 mutations per year per base pair studied. That's not a crazy estimate. That's about what you'd expect to see. Now, in larger populations, you're more likely to get the mutation simply because there's more alleles present, right? That every chromosome out there has some probability of getting the mutation. The more chromosomes you have, the more chance is that the mutation will arise. So the rate of getting a new mutation in a population might be 2n mu, right? So 2n is the number of chromosomes because n is the population size, 2 because it's diploid, every individual has 2 copies of it, and mu is that rate per chromosome. So it's the rate per year per base pair studied on an individual chromosome. Now, the mutation must also fix by genetic drift. It has to go from this rare starting frequency all the way up to 100%. So what is the probability of fixation of a new mutation in a diploid? Well, we talked about we talked about the probability of fixation of alleles by genetic drift, right? Well, let's put these two pieces together. The probability of a new mutation arising, we said, is 2n mu. The probability of a new mutation fixing will be equal to its starting frequency. The starting frequency of a new mutation will always be 1 over 2n. This is very important, 1 over 2n. Because this new mutation has arisen in the population is diploid, and there's only one copy of the new mutation. So it's one mutation in this population of 2n chromosomes. Right, so this is its starting frequency, and as we said before, by genetic drift alone, this is a probability that it'll eventually fix. Right, the probability of fixation is equal to the allele frequency. So what we're saying is the probability of new mutation arising times the probability of new mutation fixing. We put these things together, and we have a mathematical convenience. 2n mu times 1 over 2n, we can cancel these out. It's equal to mu. So this is really cool because large populations have more chance the mutation will arise, but a smaller chance that it'll actually fix by genetic drift because the allele frequency at the start is so much lower. In contrast, Smaller populations have a lower chance the mutation will arise, but have a higher chance it will fix because that starting allele frequency is high. Because of this amazing canceling out, the rate of neutral molecular evolution does not depend on population size. This was first described by Motu Kimura, whose picture is shown here. So how can we use this calculation? Well, here's an application for it. Let's say that we know the mutation rate of a particular region. Let's say we know the mutation rate for human pseudogenes is roughly 1 times 10 to the minus ninth mutations per year per base pair. Okay? So let's say we want to know the divergence time between humans and mouse lemurs. There's an interesting picture of a mouse lemur over here. So what we do is we sequence a particular pseudogene. A pseudogene, by the way, is a gene that is no longer functional. So it's assumed that mutations that arise in it are going to be neutral. They're not going to have any effect on fitness. You sequence the pseudogene and you find 150 base differences in 1,000 base pairs between the human and mouse lemur. Okay? This is not unusual. You expect several DNA sequence differences between humans and mouse lemurs. We're not that closely related. Well, we can use this to determine how far back humans and mouse lemurs shared a common ancestor. Let me show you how we do this. So again, we have this rate of 1 times 10 to the minus ninth mutations per base pair per year. Now in this case, we said we were looking at 1,000 base pairs. So our probability of getting mutations is higher, right? It'll be 1,000 times more. So we can say 1 times 10 to the minus sixth mutations in 1,000 base pairs. I just multiplied 10 to the minus ninth times 1,000. So 1 times 10 to the minus sixth mutations in 1,000 base pairs per year. And what we can do is basically just invert this. Okay? So we should say that for every one mutation in 1,000 base pairs, we can say it's been about 10 to the sixth years. I just inverted the numbers up here. Okay? So we saw 150 mutations. So 150 mutations times 10 to the sixth years per mutation. So that comes out to 1.5 times 10 to the eighth years of total divergence. This seems like it should be the right answer, right? Because this is saying this is how long we should have gotten, it should have taken for us to get this 150 mutations. The problem is there's two branches. So here's our common ancestor in time. So here's today, here's long ago. So we have this change over time. We have these 150 mutations that distinguish us. Now some of these mutations are on this lineage, but some of them are also on this lineage. So when we're saying this 1.5 times 10 to the eighth years, we're actually summing both of these things together. So what we need to do is we actually need to divide by 2. So we take 1.5 times 10 to the 8th years, divided by 2, and that becomes 7.5 times 10 to the 7th years. Or the time to the ancestor would be 75 million years ago. So as long ago we can say is roughly 75 million years ago. Okay? Take a second just to look that over, and I'll give you one to try on your own. So we start with this, mutations per base pair per year. That was a given. We then looked at how big a sequence we were looking at, 1,000 base pairs. We flipped this number around, basically from 1 times 10 to the minus 6 to 10 to the 6th. I basically just changed it so years was in the numerator and one mutation in the denominator. That made it 10 to the 6th. So for every one mutation, we have to wait 10 to the 6th years. We have 150 mutations, so we multiply that times 10 to the 6th. This is where we get 1.5 times 10 to the 8th. We had to divide by 2 because mutations are arising along both lineages. We're not looking at base differences between humans and the ancestor. We're looking at base difference between humans and mouse lemur. That's why we have to divide by 2, and therefore we get 75 million years. There's one for you to try. Here's the time what is the time to ancestor for a human to tamarin? Let's assume the same mutation rate there. Let's say in this case you screen 10,000 base pairs of sequence, okay, just so you're not using exactly the same numbers. Let's say you found 860 mutations. What would the divergence time be? Well, I hope that wasn't too challenging. Now, let me walk you through this one just like we did the previous one. So this is just filling in those same things. So we said that we're looking at 10,000 base pairs. That's the same as 10 to the 4th base pairs. So 10 to the 4th times 1 times 10 to the minus 9th. So that would be 1 times 10 to the minus 5th mutations in 10,000 base pairs. Right? So all I do is just multiply 10 to the minus 9th by 10,000. Then I invert this whole thing, so I have years in the numerator and mutations in the denominator. So we have to wait 10 to the 5th years for every one mutation in this 10,000 base pairs. Okay, so this is a longer stretch. That's why we don't have to wait as long for it. We have 860 mutations. So multiply these two together, and we get 8.6 times 10 to the 7th, or 86 million. Now again, this 86 million is reflecting what's happened in this branch and what's happened in this branch. So we divide by 2. So the time to our common ancestor would be half that, or 4.3 times 10 to the 7th, or 43 million years ago. 43 million years ago is when we may have shared a common ancestor with humans and tamarin. Now, several people have told me they're very interested in these divergence time estimates and calculating them from molecular data. If you're interested in looking up some published divergence time estimates, I refer you to this website, timetree.org. They also have a free iPhone app where you can just type in your favorite two species and see what the estimated divergence time is between them. So take a look at that when you have a chance. Now, I'd like to do a little segue into what's going to be coming up in the next set of videos. Now, nucleotide variation exists within species and between species. So let's say, for example, you sequenced a stretch from 4 individuals of species 1, 4 individuals of species 2. So this may be human and tamarin, for example. There are some bases, like, for example, base numbered 1 here, where every individual from species 1 differs from every individual from species 2, right? All individuals from species 1 have C, all individuals from species 2 have a G. So this is some sort of fixed difference, okay? We may have cases where one species is variable and the other is not, and that's what we see with both bases 2 and 3. In this case, this particular site labeled number 2 is variable in species 1, whereas it is invariant in species 2. And this one right here, this one is variable in species 2, but invariant in species 1. It's variable, but there's only one rare variant here, at least among these individuals. Now, a big question out there, we see this variation within species, we see variation between species. So between species would be these would be within species. The question is, where does this come from? Now, some mutations are advantageous, and we expect those to spread within species, and probably spread fairly quickly. Many mutations are bad, and even if they're bad, they may be still found in the population for a short period of time. You saw genetic drift in particular can allow some bad mutations to stick around for quite a while. So the question is, how much of the genome actually evolves solely by mutation and genetic drift in this purely neutral fashion, right? How much of this is actually being affected by natural selection? Well, there's two sort of schools of thought that have been around since the 1960s or so. One is the neutralist school of thought, and that is that most of the nucleotide variation that you see present within species tends to be neutral, okay? So most of the variation you see there within species tends to be neutral. In contrast, selectionists suggest that very little nucleotide variation is neutral, that if you see multiple variants, it could be something like, for example, overdominance, or it could be that particular variants are selected in this population and other variants are selected in that population, so both types stick around. How much of the variation that's out there is actually selected? How much of it is neutral? This is a very big question, and it's not something to which there are very clear answers to just yet. We'll come back to this when we start looking at patterns of molecular evolution. Hope you'll join us.